In this video, we're going to cover two topics. We're going to look at why is it that cells are so tiny, why are they microscopic, why is it that they have to stay so small, and as they grow, they have to divide to stay small. And then we'll also look at the difference between chromatin and chromosomes. Now, there's going to be some important key terms in this video. There's going to be some foundational stuff that you really need to know in order to understand the cell cycle, which is mitosis, they'll be learning about this week and in this unit. One thing you first have to understand is why cells divide in the first place. So cells grow, and as they grow, they get larger and larger, but at some point they have to stop growing or divide. And the reason why that is because cells must stay small, and that's because as they grow, their surface area and their volume ratio changes. And let's take a look at how that actually works mathematically. Look at the math real quick, just a reminder what surface area and volume is. I'm assuming you already know this. Surface area is the amount of surface that covers something, so it's the actual area of that surface that covers the object, whether it's a sphere or a cube. And the volume then is how much 3D space there is inside of that structure. So if you think about the surface area of a cell, essentially it's how much area is covered by its plasma membrane, and the volume is how big is a cell, how much stuff, how much space is there inside the cell. Now the problem as cells get larger is that it turns out that the volume of the cell will increase more rapidly than the surface area. And there's actually a mathematical reason why that is that I'll show you in just a second. So cells that are small are very, very efficient. They can supply the nutrients inside of them very easily. They can get rid of waste. They can move things from one side of the cell to the other. And so that's why cells are tiny. They're microscopic. It's because of this surface area to volume ratio issue. This picture actually shows the mathematical reason why this is. So if we take a look, let's take a cell, and let's imagine, of course, cells are not nearly this big in real life, but let's just pretend that you had a cell that's 1 centimeter by 1 centimeter by 1 centimeter, so it's a cube. Well, the surface area is simply the area of all the surfaces added together. So the area of this square is 1 times 1, and then there's 6 faces, so times 6 gives me 6 centimeters squared. That's how much surface area there would be for this cell. The volume is just 1 times 1 times 1, so that's 1 cubed or 1 cubic centimeter. So my ratio of surface area to volume is 6 to 1, right? 6 centimeters squared to 1 cubic centimeter. Now let's imagine that we take that cell and enlarge it. Instead of 1 by 1 by 1, it becomes 2 by 2 by 2. So for my surface area, it's 2 times 2, which is 4, times my 6 sides gives me 24 centimeters squared, so that's definitely increased. But now let's look at the volume. Instead of 1 times 1 times 1, it's 2 times 2 times 2. That gives me 8 cubic centimeters. So that's increased, but by a lot larger, right? This one's gone up by a factor of 4. This has gone up by a factor of 8. So now look at my ratio. My ratio has gone from 6 to 1 to only 3 to 1. In other words, yeah, my surface area increased, but look how much faster my volume increased. And the reason why this is, if you think about it, if you think about our math, a squared function, right, it looks like a parabola kind of like this. A cubic function is steeper, right? The curve gets faster quicker. And this is what they deal with cubed functions. They get bigger faster than squared functions. Let's look at it one more time just to kind of prove to ourselves that this is really the way things work. Imagine we stretch the cell out. It's bigger now, 3 by 3 by 3. So the surface area is 3 times 3 times 6. That's 9 times 6 is 54 centimeters squared. The volume is 3 times 3 times 3. Now we're all the way up to 27. So this is increased by a factor of 9. This is increased by a factor of what, 13 and a half? So now my ratio is down to 2 to 1. So what this essentially means is as cells get larger, they have way more volume than they did before. And so it becomes harder for diffusion to allow things into and out of the cell and through the cell as easily. That means that smaller cells are more efficient and they function much better, which is why cells are so tiny. That's why they're microscopic. So as cells grow, they really at some point reach a limit where they're getting too big to function properly and they have to either stop growing and stay a certain size, or they divide into 2 smaller cells and then each cell can keep growing. So substances that are transported within a cell, they can move by diffusion or motor proteins, and they move much more efficiently in a smaller cell. Now, if you were in front of me in a class, the example that I always do for this is I take a balled up piece of paper and I pick a student at a desk and I say, okay, you, let's say Jonathan, you're sitting at this desk. You're the nucleus that's controlling the cell. The cell is the desk that you're sitting at. So the kind of borders of the table are like the cell wall. So this crumpled up piece of paper represents a ball. So let's just kind of draw it out here. So we got Jonathan, and he's sitting at his desk, and the crumpled up piece of paper is now right here on the edge of his desk, and I say, okay, Jonathan, why don't you go ahead and grab that and bring it into the cell. And of course, Jonathan very easily reaches out, grabs it, happens very quickly, happens very easily. And then I say, okay, Jonathan, now you're still the nucleus, but now instead of your cell just being your desk, now your cell is the entire classroom. So the walls, the ceiling, the floors, these are now the cell walls of your cell. And here's the paper, and I walk all the way over far away from Jonathan. I say, okay, that piece of paper, that crumpled up thing that you want is right here. And I make Jonathan get up. He has to walk all the way over, grab that paper ball, bring it all the way back to his desk. And of course, that takes longer, and it takes him more energy. So he's much faster and more efficient when his cell is smaller versus when his cell is larger. So go ahead and explain in your own words the example on your study guide there. There's actually a lab that some teachers do. I don't. I don't think it's a great use of class time as far as the amount of time it takes. But essentially what they do to kind of show how this works as far as volume increasing much faster than surface area is they take these agar cubes and they have the students cut them. This is a 1 by 1 by 1, 2 by 2 by 2, and a 3 by 3 by 3 cube. And they put that in a solution that then will dissolve into it. And you give it a day, let's say, and then afterwards what you see is that in the small cube, because there's more surface area compared to the volume, compared to these other ones, that pink dye diffuses all the way into the cube. In the larger cube, you can see it's dissolved the same amount, but because there's so much more volume, it hasn't reached all the way to the center, so it's not very efficient, and then especially in this larger one. So again, this just helps us to illustrate this problem that cells have with surface area and volume, that as they get larger, yes, the surface area increases, but the volume increases much faster. When we talk about the cell dividing, we have to really kind of address this issue of chromatin and chromosomes. So understand both chromatin and chromosomes are DNA. Chromatin is the relaxed, string-like, usable, make a note, this is sort of the DNA form when it's being used, form that DNA can come in. It's sort of when we draw these pictures of DNA, what we're really drawing is DNA in its chromatin state. It's relaxed. Think of it like a rubber band that's been relaxed and it's just kind of laying there. Now take that same rubber band in your hand that's relaxed and start to twist it. And what happens is as you twist it, it forms these little twists, obviously. And then if you actually do it with me right now, what you'll see is that it twists some more and more, and the twists start to twist on themselves, and it kind of bundles up, and it gets shorter and more kind of compacted. That would be like the chromosome DNA. So what chromosome DNA is you take this chromatin DNA and you compact it. And you compact it, twist it, compact it, compact it, compact it. And now you've got your chromosome. Now I want you to understand the DNA is only in the chromosome state when the cell is going to be dividing. Make a note of that over here. It's in the chromatin state most of the time, and it's in the chromosome state when it's dividing. And the reason why that is because the mother cell, that's what we call it. We call the original large cell that's going to divide into two cells, we call it the mother cell, and it's going to divide into two daughter cells. So there's no father and son cells, we call them mother and daughter cells. So that original large cell that's grown and is now going to divide, it wants to take the DNA that it has, which is represented by this chromosome, and it wants each of its daughter cells that it's going to split into to get the exact same copy. So what it'll do, and we'll learn about this later on, it's going to copy all of its DNA, and then it's going to take this chromosome and attach it to its identical copy, and we're going to call that a pair of sister chromatids. That'll be in your notes in just a second. And those sister chromatids then are used to separate, and as they separate then into their two daughter cells, the mother cell will know that she is going to be sort of dividing evenly, and that both of her daughter cells will end up looking exactly like she did. So this will make a little more sense as we actually look at how the cell mechanically divides later on. For now, just understand the chromosomes are this compacted DNA, and it's used when the cell divides only. This picture illustrates it beautifully. You've got 4 cells in this picture. 3 of them are not dividing. You can see the nucleus quite nicely outlined. You can even see the nucleolus inside of it. And then the DNA, you can't really make it out because it's this thin, these tiny little strands. This is the chromatin state of DNA. This cell is dividing. We'll learn it's in a phase that we're going to talk about later called prophase, which is sort of the first step of the cell dividing. And you can tell it is dividing because notice the DNA has compacted into these chromosomes. We'll also see, notice the cytoskeleton looks very different. We're getting this what we call spindle apparatus. the centrioles are forming the spindle apparatus. See, it looks very different than the cytoskeleton of these cells. So it's very easy to tell when you look at cells whether they're just growing or whether they're actually in the process of dividing, whether we're in the chromatin state or the chromosome state. So there's another term here we'll be using in just a minute here as we kind of are looking at mitosis and diagramming it. Sister chromatids. So this is one chromosome. This is its identical copy, and together now we call them a pair of sister chromatids. Another term that we'll be using a lot as we're diagramming this is the centromere. The centromere is the location specifically where the sister chromatids are actually attached, where one chromosome is attached to its identical copy, making a pair of sister chromatids. That location is called the centromere. Here's another picture that shows with the fluorescent microscope, you can see all the different states. We'll learn about the names of these later. We don't have to worry about that right now, but I just want you to realize that, when the cell's just kind of sitting there growing, copying its DNA, that DNA is in the chromatin state, and you can see that it forms these chromosomes which pair off into these sister chromatids, which are then separated, right, into the two daughter cells that are going to be forming. So this will make a little more sense as we kind of illustrate it in class later on, but for now I just want you to realize that there's this link between chromatin and the chromosomes, and then the sister chromatids, where you have those chromosomes attached together, and then when they separate, we know that each daughter cell is going to get that original chromosome just like the mom had. Before we start actually diagramming mitosis, the last thing I want to talk about is just sort of chromosome number. So in the somatic cells, which are the body cells of a human being, so all the cells essentially except for the sperm and egg, depending on if we're talking about a male or a female, right? So all of the rest of those cells, the heart cells, the liver cells, the kidney cells, the skin cells, the brain cells, all of those other cells are going to have 23 matched pairs of chromosomes, 46 chromosomes total. Okay, so this is for a human being. You've got 1, 2, 3, 4, 5, all the way to pair 22, and the 23rd one would have a number because it's kind of a special one that determines our gender, and so that's our 23rd pair. So we have 23 pairs, and each of those pairs has 2 chromosomes. So in human beings, we think about how many total chromosomes we have 46, right? 2 times 23 gives us 46. There's 46 individual chromosomes here in a pair of what's called a homologous pair, 23 homologous pairs. Now, that's not the case for all organisms. So for humans, it's 46. For dogs, actually, it's 78. Tigers, it's 38. So this is what it looks like. By the way, this thing is called a karyotype. That one that was on the last slide is called a karyotype as well. So you can see for dogs, like our dog at home, we've got, dogs have 38, 39 pairs. So not 23 like us. They have 39 pairs. So 39 times 2 gives us 78. Also, another little side note here, in this case for the dog, the 39th chromosome pair is, again, those sex chromosomes that determines the sex or the gender of the dog in this case. An X and a Y is a male, an X and an X is a female. So dogs have 78 chromosomes in 39 matched pairs. Tigers, they have 38 total chromosomes. How many matched pairs do they have? They've got 19 matched pairs, right? 1 through 18, and the 19th is the sex chromosomes again. And for a tiger, what gender tiger are we looking at here for this karyotype? Well, female, X and X. So in humans, we have 23 matched pairs for a total of 46 chromosomes. So 46 chromosomes total in 23 matched pairs. That 23rd one again, is this a male or a female we're looking at? It's a male, XY. That's that the 23rd pair is the sex chromosomes that determine the gender. So now all that background information is kind of under our belt, let's look at how the cell actually works, how the cell cycle works. And essentially what we're looking at is the growing and the dividing of cells. It helps keep the cells from getting too large, which allows them to stay efficient. And actually on a multicellular, make a note, this is for multicellular organisms, it allows them to grow and heal. So the reason why you are as large as you are, remember, if we think about it all the way back to the beginning, you started life as one egg, right, meeting one sperm, that became one fertilized cell that divided into 2, which divided into 4, and then into 8, and so forth, right? And so you are larger than the elementary school students around this campus, and you're larger than my toddler 3-year-old son because you are made of more cells. So the increasing the number of cells allows for multicellular organisms to grow. It also allows you to heal. When you have an injury, let's say a cut on your arm, and that gap now, so right, you get your skin, and then you get a cut in that skin. There's actually a gap there that's missing in your skin. And the way that it heals is the cells on either side are going to start to divide and fill in that gap, and then you get that healing done. So it also allows us, these cells growing and dividing, is going to allow the organism overall to grow and heal as well. So I hope this video was helpful introducing mitosis, and we will see you next time.